Thank you, Susanna. It's great to be here. It's great to tell you a little bit about what's going on in my lab at Stanford. Let me see if I can find the clock. Where can I? Okay. Thank you. Great. So I'm in an earth science department at Stanford, and all of my degrees are in earth science department. So my orientation towards understanding microbial ecology is very much rooted in how are they affecting global processes, biogeochemical cycles throughout time. And there's a couple of reasons I'm interested in this. To name a few, I want to explain climatic events in the history of the earth. I want to help the search for life on other planets. And I want to be able to predict changes in climate on Earth. So there are a few questions that I focus on to get to these bigger topics. Who does what in situ? What influences who does what and how much? And will these metabolisms act as a positive or negative feedback to climate change? To get at these, we have to bridge huge spatial scales. And I think many of us in this room focus on the really tiny side, including individual biomolecules, and you're going to hear a lot about DNA and RNA in this talk. But I'm also going to talk a lot about what happens at the single cell level. And one of the instruments we use to do that is nanosims. And thinking about how we can build up from here into bigger, more global scale models is one of the things that I'm ultimately interested in. So I primarily study the marine environment, and for those of you that don't, this is just a little bit of an introduction to that system. About 30% of all of the cells, the microbial cells in the ocean, are in the top euphotic zone, so here we have a lot of photosynthetic activity. But the majority of cells in the ocean are in the dark middle of the ocean, where you still have oxygen, but you have no light. And then 15% of all the cells in the marine environment are actually in the top 10 centimeters of marine sediments. So you have extremely densely packed cells in marine sediments. And in each of these areas, you have very distinct microbial metabolisms and interactions that are processing or indirectly processing molecules that are relevant for the climate, like CO2, methane, and nitrous oxide. And I'm interested in understanding who produces what and what controls those particular gas cycles. My lab focuses on the dark ocean, so the water column and the sediments. And we're going to be walking through about 4 different topics today in the next 30 minutes. So one of the primary questions is, do Thaumarchaeota assimilate organic matter? And to get there, we're going to first touch on how we can do high throughput screening of metabolic activity, which is, I think, a question that no matter what system we study, is probably relevant. Then we're going to go down to the sediments, and the primary question we're going to answer there is who fixes nitrogen in deep sea sediments. But again, before we get there, we're going to talk a little bit about the diversity, function, and novelty of these microbes in marine sediments. So let's start with the water column. To start with a very basic question, what percentage of cells are active in the deep water column? This actually isn't a very easy question to answer in this system or in any system, because if we look at DNA or RNA, we might get an idea of the metabolisms that are active, but we don't know really what percentage of the cells are mediating them, how many of them are actually active. So together with the group that I did my postdoc with at Lawrence Livermore National Lab, we developed an approach for this using multiple stable isotope incubations. So here you can see if you take a container of seawater and incubate with both 13C labeled bicarbonate and 15N labeled amino acids, but it could be even ammonia, something that anything could take up. You have some cells that take up one, some cells that take up the other, and some cells that take up both substrates. And based on this, we can call them autotrophs, heterotrophs, or it's complicated. And I have it listed there as a mixotroph question mark, because for the purposes of today, that's really all I can get into, but because of recycling and anaplerotic reactions, it actually depends a lot on the ratio of which of these to kind of parse it out further. But what we get from this is a fraction of the cells that are active, as well as the fraction that are doing each of these kind of broad metabolism categories. We analyze this in the nanosims at the single cell level, so we've already heard a little bit about nanosims this morning, so I'm not going to do a full introduction to it, but basically we get isotopic composition at a 50 nanometer spatial scale. So here you're looking at a a scan, you usually don't hear the phrase high throughput and nanosims in the same sentence, but in this sense we actually did, and this is because we haven't identified which cells these are. These are just cells filtered on a 0.2 micron filter, and then we're scanning them. So in the course of 12 to 24 hours, you can analyze hundreds and hundreds of cells. So each one of these bright little dots, which is shown here as a CN, on a CN ion scale, shows you where an individual cell is. I want you to focus on these 4 individual cells for the next few seconds. You're now looking at the 13C, 12C ratio of all of those same cells, and you can see many fewer of them are visible now. And of the 4 we were looking at, only 2 of them are enriched in 13C. Now you're looking at the 15N, 14N ratio, and a different 2 of our 4 cells are enriched. And if we go back to our original map of where all of the cells are, we can then classify each of these cells individually based on their metabolism. We can sum these up to get a fingerprint of the community. We see about 90% of the cells in this incubation were active over the 24 hours that it was incubated for. And using some kind of creative inhibitors, we can also start to see trends. So in another incubation, we incubated with bacterial antibiotics, ampicillin and streptomycin, to enrich for archaea. We confirmed with sequencing that we did enrich for archaea. And here we can see much more incorporation of the 13C bicarbonate. So using this approach kind of on a wider scale, a postdoc in my group, Nestor Arandia Gorostidi, used this approach at a whole range of water depths in the water column, and additionally asked the question, how are they getting their nitrogen? How does organic nitrogen play into the metabolism of deep sea microbes? So what you can see here is, in addition to seeing the percentage of cells that are enriched in one substrate or another, and to the person's point who asked in the previous talk, how do you know it's direct consumption of this substrate? We don't. So we think of it as urea-derived nitrogen or ammonia-derived nitrogen. In that case, it's probably ammonia. But in addition to seeing the percentage of cells that are incorporating each one of these, so in the case of ammonium and amino acids at the surface layer, it's about 90% of cells are incorporating either one. You can also see the histogram or the distribution of activity, and you can see that it's quite different for ammonium and amino acids. For ammonium, it seems like most of the cells that are enriched are barely enriched, but for amino acids, it's much more spread out. And this is the kind of view we get when we take this single cell approach. So you can see how this works throughout the water column, and in general, the cells become less active with depth. That could be due to more dormancy. It could be due just to the fact that they're growing more slowly, so we haven't captured it during the time of our incubation. But one thing I want to point out in particular is at 150 meters, you kind of have a spike in this urea utilization. So again, we don't know necessarily if 75% of the cells are actually assimilating urea, or if we have a much smaller group of cells that are very effectively kind of unlocking that resource for the rest of the community, who are then assimilating secondary products. But you do see that at this depth, you have more access to urea than you do elsewhere. When we look to see who is in the community at that point to get additional clues, you can see that this is actually where the Thaumarchaeota begin to spike in the water column. So they're this peach color here, and we think that they are likely involved in this unlocking of the urea, and you're going to hear more about that in a second. So let's talk about the Thaumarchaeota specifically for a few minutes. You probably don't think that much about archaea if you are a regular person, because when you go swimming, you're only swimming in about 1% archaea at the top of the ocean. The fact is, the deeper you go down, the more and more relatively abundant they become, until in some locations in the ocean, they actually equal, if not exceed, the concentration of bacteria, which is huge. It ends up being approximately 30% of all the microbial cells in the ocean are archaea, and about 80% of those are this single phylum, the Thaumarchaeota. They were previously called marine group 1 Crenarchaeota, if you've heard of them as that, and in the future I hear their name might change, but today I'm going to be calling them the Thaumarchaeota. Based on the few pure cultures we have and a few other data sets, which I'll talk about more in a few minutes, we believe they are ammonia oxidizers, aerobic ammonia oxidizers, and capable of fixing bicarbonate for their carbon. They are also the ocean's biggest source of N2O. There's conflicting evidence over whether or not they take up organic matter, and this is what I'm going to be focusing on for the next part of this talk. So there's very good evidence that they are capable of taking up inorganic carbon. That comes from culturing work, that comes from genomics, that also comes from some stable isotope tracing experiments. But there's also a lot of evidence that they're able to take up organic matter. That also comes from genomic data sets, some density gradient stable isotope probing, a lot of marfish, specifically looking at their ability to take up amino acids, and a really cool radiocarbon study that showed if you look at the radiocarbon of deep sea archaeal lipids, about 20% of them should be attributed to organic carbon as opposed to inorganic carbon. But when you do these bulk analyses, you don't know if a single cell is doing 20% and 80%, or if that's split over the whole population. So you can imagine there's a few different scenarios. You have the 20%, 80% in separate cells. You can have all mixotrophs, so they're all doing both. They're all getting some organic and inorganic carbon. You could have a combination of this, and you can have different kind of ratios of that combination. So we can use a single cell approach to try to parse these apart. And I should mention, the reason we really care about this is because each one of these scenarios, whether they're acting autotrophically or heterotrophically, has different implications for the cycling of the greenhouse gases we care about, the CO2 and the nitrous oxide. And I think it also affects what we might see in a changing ocean as our conditions change, which we might see more or less of in the future. So to get at this, we collected a natural sample at the San Pedro Ocean Time Series, which is off the coast of Los Angeles. We focused at 150 meters water depth because you often get the highest diversity of Thaumarchaeota there. It's kind of a mixing between the shallow groups and the deeper groups. We did metagenomics and metatranscriptomics, and then 13C and 15N incubations, followed by fish nanosims. So I don't think I need to explain MAGs to this group, but my postdoc Alma Parada kind of led the omics side of this project. We were able to recover several Thaumarchaeota bins or MAGs, and in them we looked for the pathways that were responsible for inorganic and organic carbon. So when we break out our contigs into specific bins, we can see that yes, we have evidence for both organic and inorganic carbon and nitrogen assimilation, similar to previous studies. We see that within individual bins, we have both gene sets available, and we see that these were also transcribed in our sample that we collected right after, or that we filtered right after collection, suggesting that they're transcribing both in situ, maybe they're living mixotrophically in situ. Now we want to do the isotope probing experiment to see what we can see directly in the individual cells. So we're going to incubate the water with organic nitrogen and carbon and see what they're able to take up. So many of these substrates are intermediates in the TCA cycle, which we thought would be a good place to start, in addition to other kind of commonly assimilated substrates in the marine environment. Fish are fluorescence in situ hybridization, as opposed to the high throughput method where we're quickly scanning but we don't know who the cells are. Fish allows us to know exactly which cells are Thaumarchaeota before we get their isotopic composition, so we can be sure we're looking at this particular group when we get our isotopic value with the nanosims. And then the nanosims is what will give us the isotopic value. These measurements were done at Lawrence Livermore National Lab. So beginning with amino acids, first let me orient you. On the y-axis, we have our nitrogen uptake. So in the case of doubly labeled substrates, this is the nitrogen of that doubly labeled substrate. And on the x-axis, we have carbon. In the red, we have the Thaumarchaeota cells. In the blue, we have bacterial cells. So what you can see here is although they're taking up the nitrogen, they're not taking up the carbon. Interesting. We see something different with urea. We see much more nitrogen uptake, but we do also see a little bit of carbon uptake, just a little though. And the proteins, we see uptake of neither the nitrogen or the carbon. So some interesting things are going on with the nitrogen, but the carbon story is kind of clear. There's minimal organic carbon uptake by this group, at least by these 3 substrates. Then we switch to our purely carbon substrates. So now we have 13C labeled carbon substrates plus 15N ammonium. And the 15N ammonium is serving as a general indicator of activity. So we can see if the cell is at least anabolically active before we assess whether or not it's taking up that 13C. So from pyruvate, glucose, and oxaloacetate, we see the same thing. We see active cells who are not assimilating the organic carbon. Interestingly, the 15N incorporation from ammonium is often higher in the archaea than it is in the bacteria, which is a little surprising because most people think of them as growing more slowly, yet they have this much higher 15N incorporation. So to focus a little bit more on urea for the moment, we saw both 13C and 15N uptake. This is consistent with what other groups have shown, that the Thaumarchaeota are capable of cleaving urea with urease, and this is actually a source of ammonia for nitrification, their catabolism. What we're showing here is that they're also using these molecules for biomass. And when we look into our metagenomes, we do have the genes in the pathway that would be necessary for this. Here you're looking at the UREC phylogeny, so the genes are present and they're also expressed in our sample. So to summarize that section, in our omics analysis, we do see widespread genetic potential for organic matter uptake within the phylum. And even within individual mags, we see both of these capabilities and even expression in our in situ sample. But we see something a little bit different when we look at the stable isotope probing experiments. We don't see that organic carbon is a primary carbon source. We see that some Thaumarchaeota are capable of taking up urea and amino acids, but it's much more nitrogen than carbon. So if I were to kind of summarize this visually, we have primarily autotrophs here. There might be some members of the community, and you could see probably a few cells that weren't kind of following what the rest were doing. There could be intraphylum heterogeneity, but primarily they seem like they are autotrophs, but they're using organic matter primarily for nitrogen. All right, switching gears a little bit, let's bring sediments back into the picture. My group was lucky to go on a 9-day cruise about a year ago off the coast of San Francisco, where we collected samples along a 350-kilometer gradient transect. What this allowed us to do, these were collected by Multicore, by the way. What this allowed us to do is look at the diversity and activity of microbes down to about 30 centimeters, and in a couple cases much deeper than that, 2-meter cores, over a whole lot of different physical and chemical conditions. And I'm not going to get into all of the details of this because my postdoc Alma has a poster on this tonight, which I encourage you to go see, but I do want to give away 2 kind of interesting tidbits or teasers, which you can ask her about. One is that when we look at the diversity or the 16S sequences in all of these samples, we see that the novelty increases with sediment depth. And the majority of the cells we're detecting are novel. So on the x-axis here, we have an average percent ID, which takes into account both the percent identity to a 16S sequence in a reference database and how abundant that particular ASV is. And you can see on average, especially if we put up thresholds that give you some idea of where the novelty lies, most of the organisms are novel at least the genus level, and in some cases much more so than that, and this increases with depth in the sediment. So we're really excited about looking more closely at some of these very deep microbes to figure out who they are and what they're doing, and potentially what some of these interactions are. In order to get at that, we've been collaborating with a group in the medical school at Stanford that uses a read cloud assembly approach to basically make mini metagenomes out of the enormous complexity of the marine sediment metagenomic world. And this has helped us get more complete bins and also ones that include 16S. And so you can talk to Alma more about that if you're interested in that approach. We're also excited to be collaborating with the JGI with this sample set in the coming year to hopefully know more about these microbes. And so maybe in future user meetings, I'll be able to tell you more about exactly what these novel groups are doing. All right, switching gears again, we're going to stay in the sediments, but talk specifically about one particular metabolism, nitrogen fixation. So a little bit of background, nitrogen fixation is the biggest source of bioavailable nitrogen to the biosphere. Deep marine sediments are usually not thought to be a huge source of nitrogen, primarily because they're thought to have plenty of nitrogen available, so you wouldn't need to fix nitrogen. The truth is that's not true, especially at the very top of sediments. You often see low levels of nitrogen that would not normally inhibit diazotrophs, for instance, in pure culture. And there's more and more evidence that shows that there is active nitrogen fixation in at least some areas of the deep sea floor, particularly with elevated carbon loading. So our question is, who are they? We actually have very little information on that at the moment. From a few different lines of evidence, we can see that there's a few methanogens, methanotrophs, and there's kind of indirect evidence of sulfate-reducing diazotrophs. But we feel like we do not know the full diversity. If you do a NIF-H analysis, there's a ton of different NIF-H sequences. We don't know who they belong to. So in this part of our work, we're trying to figure out who's doing it. We used one particular sample that's from the very end of Monterey Canyon. It's almost 3,000 meters water depth, so approaching a representative depth of the full ocean. In a paper we published just recently, you can see in both the 0 to 3 and 9 to 12 centimeter depth horizons, we saw active nitrogen fixation through 15N2 incorporation, at least on a bulk level. And we wanted to investigate these two horizons to see who was responsible for it. So we do incubations with 15NH4 as a general proxy of activity, and 15N2 is a specific indicator of nitrogen fixation with and without methane at these different depths. This is work that's being led by my graduate student, Bennett Capilli. Extracted the DNA. He developed a pipeline to identify the 16S, or the phylogeny of the NIF-H host organisms, as well as doing density gradient SIP, which we heard about from Sam, who's actually a collaborator on this project, to get at this question. So first, looking at the NIF-H sequences, this is a tree showing with the purple branches where our NIF-H sequences fell, given the entire database, and you can see they're extremely well distributed. We detected 434 unique ASVs total in only these two different samples. And you can also see that most of them are clustering or identifying with delta proteobacterial NIF-H sequences. But we do have a greater diversity in addition to that, including the firmicutes, the acidobacteria, and a few other groups. When we look at the SIP data, we see a similar diversity. So based on that stable density gradient separation that Sam described, when you look at the heavy fractions and sequence just those to see who has incorporated the 15N into their DNA, we see that it's primarily delta proteobacteria, consistent with the NIF-H results, but there's a bigger diversity there too. And what's nice about this approach is that you don't have to be targeted with it. So if you do a fish nanosims kind of approach to figure out who's fixing nitrogen, you have to have a very specific hypothesis. This gives us a broad overview, and you're also not limited to your database of NIF-H sequences. So for instance, we're kind of excited about this peak that you can see in the planktomycetes. This is not a group that's been shown or known to fix nitrogen, but just a few months ago, there was a paper in Nature Microbiology that showed in a nearly complete mag that a planktomycetes did contain the full complement of NIF genes necessary to make nitrogenase. So it seems like there could be something interesting there. Overall, the two data sets were consistent, so we saw similarity between the groups that they were suggesting were capable of fixing nitrogen, particularly some of the ones I've already mentioned, the firmicutes, the acidobacteria, and the gamma proteobacteria. There's false positives and false negatives in each of these types of data sets. So it's really nice to be able to compare them to see where we see the consistent results. If we look up the known metabolisms of the closest relatives to these organisms, which is, I realize, is a few steps away, because nothing is that closely related to the specific organisms that are living in the deep sea that we usually have in our reference databases. If we do that as a thought experiment, we can see that actually the metabolisms associated with these microbes are very diverse. So we see them using a lot of different electron acceptors. Some of them are kind of what we expect because we had a hypothesis that sulfate reducers would be involved in this process. But we also see evidence for organisms using iron, and I don't have it up here, but oxygen and even nitrate as an electron acceptor. So they might not be doing this at the same time that they're fixing nitrogen, but it shows that there's a lot of catabolic diversity potentially paired to diazotrophy in the deep sea. So to summarize this part of the talk, from our NIF-H analysis, we can see that there's phylogenetically diverse diazotrophs, primarily delta proteobacteria, but other groups as well. Our 15N DNA SIP experiments showed us that the putative diazotrophs include delta proteobacteria, as well as several other groups. And the diazotrophs are potentially catabolically diverse. And the reason this ultimately really interests me is I think what it tells us is that the deep sea kind of has this capability to access the most abundant form of nitrogen on the surface of the Earth, N2, when it needs to, under all different geochemical conditions, as conditions change, because the diazotrophs can pair it with all these different metabolisms. So yes, it's happening in some places now, which we can measure, and other places it might not be happening under current conditions, but the potential is there. So as conditions change, this metabolism would be accessible to them, and they could overcome nitrogen limitation. So to come back to my original questions, I'll give you the quick summary of all of these. So for high throughput screening of metabolic activity, we saw that you can do a multi-isotope, stable isotope labeling experiment followed by scanning nanosims approaches to get fractions of active and inactive and broad categories of metabolisms in a community. I think this approach is particularly useful for communities with extremely low biomass, where it's hard to get enough DNA or RNA to look at it that way. It's also useful in communities where you don't know how to interpret that sequencing data. If you don't know what an organism does because we've never seen it before, then it's less useful to have it in your genomic data set and maybe more useful to see an actual evidence of activity here. From the Thaumarchaeota question, do Thaumarchaeota utilize organic substrates? We saw that primarily they don't use organic carbon. We do see a lot of use of organic nitrogen. We think this could be because they're breaking it down and using the ammonia for their catabolism, the ammonia oxidation, which explains why they have all of the genes necessary for taking up this organic nitrogen. From the diversity and function of novel microbes, I encourage you to go see Alma's poster this afternoon or this evening, but the takeaway there was that the novelty increases with sediment depth. And in terms of who is fixing nitrogen in marine sediments, we can see that they're phylogenetically and likely catabolically diverse. So I'd like to thank my group at Stanford, as well as my collaborators at Lawrence Livermore National Lab, Xavier Mayali, Jennifer Petridge, and Peter Weber, Jed Furman at the University of Southern California, and at Caltech, Victoria Orphan, Gray Chadwick, Stephanie Condon, and Abigail Green Saxena. Thank you.
Hi, do you have to worry about pressures for your SIP experiment for growing these deep sea microbes?
Right, yes. So the pressure in the deep sea could be 400 times that what it is at the surface, at the depths that we're looking at. So we have to accept that given we're not doing high pressure incubations, we do our incubations at 2 times atmospheric pressure, but again, that's nothing compared to 400 times. We have to accept that probably what we're seeing is a subset. I don't think that an organism would fix nitrogen if it couldn't at the wrong pressure, but I do think we're probably missing some. And most people that study the effects of pressure on organisms think that around these depths, they're still only facultatively barophiles, and the really obligate barophiles don't kick in until you go deeper than this. So I think we're probably getting something that's pretty representative, but likely not complete.
Hi, Anne. So my question is, in terrestrial environments, often the whole nitrogen fixing module can be horizontally gene transferred. Do you have any evidence that's happening with the deep sea guys?
So we haven't looked into that directly. In general, we have been thinking about horizontal gene transfer as an enemy, not an opportunity to investigate, because of course the horizontal gene transfer makes trying to figure out who the NIFH sequence belongs to much more difficult. And a lot of Bennett Capilli's time has gone into developing this pipeline that minimizes potential false positives due to horizontal gene transfer, and basically we just don't identify a lot of our sequences because of issues. They're in places of the tree where it is kind of too close to call. And so of those 440 ASVs, we found that about 50 of them were in these too close to call cases. So in one way, maybe I could answer your question like that. We kind of were frustrated that, oh darn, there's a chance of horizontal gene transfer, so we can't identify them. On the other hand, yes, that is evidence perhaps that it's happening, and we could look into it more from that perspective.